and thank you for joining us for this evening's TI Technology webinar hosted by Texas Instruments, where we will take a look at breast cancer when good DNA goes bad. I see that many of you are joining us for the first time tonight, so we'd like to offer a special welcome if this is your first T-Cubed Professional Development webinar. We'd also like to thank everyone for being here with us tonight. My name is Stacy Thibodeau, and I will be the moderator for tonight. I have been a science teacher for the past 23 years. I currently teach at Southside High School in Youngsville, Louisiana, where I teach all levels of chemistry 1 and 2, and an introduction to robotics class where I use TI technology. I use TI technology to assist my teaching, data collection, and modeling math concepts, linking them to the science concept, as well as the TI Innovator Hub for my robotics program. Tonight, we have the famous Jeff Lucan here with us. After a 35-year high school science teaching career, Jeff retired from his own classroom in 2014. Since then, he's been working with science and math teachers on implementation of TI technology in their classrooms. One of his passions is the true and seamless integration of science and math in a way that benefits both curricular areas. In addition to the on-site classroom work, Jeff does presentations at state, regional, and national conferences. Jeff has been my mentor since 2014. I am so happy to be working with him tonight. Thank you for being with us, Jeff.
Well, thank you, Stacy, and I'm happy to be with you as always.
If you have any questions tonight, please use the chat window. Between Jeff and myself, we will definitely answer any questions and pause if we need to answer those questions. Jeff, you want to go over the agenda real quick?
Love to. And I'll be more specific about this particular activity and the topic we're doing, because to be sure, it's not the norm for these webinars. We're doing something very, very different tonight, and to highlight a topic that we're coming up on a month in October when we do have Breast Cancer Awareness Month. But another thing that we're doing tonight is showing some of the power of the TI-Nspire that you may not have thought about before, and we're going to do it through the story of a woman named Dr. Kristi Egland, and I'll tell you more about her in a little bit. Dr. Egland is a researcher at a local research facility in the town that I live in. So we'll talk about her. We'll talk about her journey with breast cancer, and so she'll be kind of the center of attention tonight. And then the way this is going to be a little bit more atypical of a Tuesday or Thursday night webinar is we're going to be doing quite a bit of biology. And even though we're coming up on Breast Cancer Awareness Month, which is awesome, awareness is great, but education is even better. And learning about the ins and outs of how things really happen, rather than just simply being aware of them, might be of value to, hopefully, to all of us. Some of the amazing things that the device, the TI-Nspire, can do, and the TI-Nspire software can do, are things like simulations, and we'll watch the simulation of the growth of a tumor in this activity. And then we'll actually talk about the root cause of cancer, which is the DNA. Something happens with the DNA, which is inside the nucleus of your cells. And that's, we'll talk about DNA replication, and we'll take the DNA replication challenge. Actually, I'll take it, and I will completely embarrass myself because I'm not very good at it, but it'll show you some of the really cool stuff that TI-Nspire can do. And then Stacy tells me at the end of this thing, we are going to have a drawing and somebody's going to win some cool stuff. Stacy, is that right?
That is true. A participant will win their choice of a handheld TI-Nspire or a TI-84CE. So stick around for the end. some things that Jeff wants to make sure he covers as well. He mentioned obviously some information about the simulations. He wants to also go through some different strategies and making sure that you guys have a full understanding of the DNA concepts. I did mention we're going to use the WebEx chat. Please make sure you send any messages that you need. Jeff and I, like I said, are going to go back and forth, making sure we can see the chat and answer any questions. But at the same time, if there are things you want to comment on, please feel free to do that. We would like for you to share what you're learning today using the @TI calculators or hashtag T3Learns. Both of those will get you on any social media platform. And then that just gives you that opportunity to reach out. Tonight, the session is being recorded. So if you want to follow along on your handheld or your teacher software, please feel free to do that. At the end of the session tonight, you will get a link for any documents that Jeff shares with you, and as well as the information will be sent to you in a couple of days. All right, Jeff, I'm going to stop sharing and you can share your screen.
Awesome. Stacy, thank you. And I have a couple of different screens that I'll be sharing, or a couple of different items I'll be sharing with you. Hopefully you're able to see these. Before we just jump into this activity, because it will spend our entire time tonight going through this activity, I want to show you where you can find it. Now, if you are a groupie with the webinars, which I know some of you are, you attend on a regular basis, and even if you're not a regular attendee of the webinars, if you're new to this because you saw the subject tonight and you're like, man, I want to learn more about that, or I'm curious about that, I want to show you where you can locate this activity. If there's any website for your science and or math or STEM teaching that you might want to just memorize or bookmark it in your browser, it's education.ti.com. Everything educational with TI can be found on that website. Now, where we're going to go to find this activity, and I'm just going to show you where it is, and then I've already opened it up, so I'll show you the activity in just a moment. But I think it's pretty self-evident. If you go to activities, I'm just hovering over the activities tab. This particular one is under what's called STEM activities. If you're a math teacher and you use TI-Nspire, you've probably spent most of your time on this particular pull-down menu on math inspired. If you're a science teacher, maybe science inspired, which are specific, or let me back up. They are activities specifically made for TI-Nspire. The STEM activities is where we're going to go for this this particular, not only this individual activity, but group of activities. So here we have the Inspiring Generation STEM splash page. I'm just going to scroll down a bit because it is down here just a little bit, a little ways. STEM Behind Cool Careers is another area that we have some activities. The area we're going to be exploring tonight and the activity that we're going to be using is found in this area called STEM Behind Health. I'll give you a little more background about that in just a moment and where that idea came from and the kind of the history of this particular area of the TI website, STEM Behind Health. Many of you are probably familiar with the STEM Behind Hollywood series. In fact, we're doing another webinar in October, I believe, on one of these activities out of the STEM Behind Hollywood genre, which is called the Zombie Apocalypse. So you're not going to want to miss that. It's October 20 something. I can't remember the date, but we'll be doing that one as well. The STEM Behind Health actually came after the STEM Behind Hollywood. So I'm going to click on Learn More. And again, what I'm doing here, if you're just joining us, is showing you where you can find this activity on the TI website. So here's our STEM Behind Health splash page, and there's several activities here that we've developed. The first one listed as you scroll down is called Breast Cancer When Good DNA Goes Bad. to access this activity, we would click on see the activity, but I want to make sure that I show you that there are some others here as well. There's one on diabetes research, and one of the more popular ones that we've done is on type 1 diabetes. This one actually makes use of and tells the story of a former student of mine who was diagnosed with type 1 diabetes when she was 12 years old. But that's another show. We're not going to talk about that particular activity tonight, but I just wanted to show you that there's more than just the breast cancer activity that's available. I'm going to click on see the activity, and whenever you click on you want to see what's available for the activity, there's going to be several files that are available for you to download. The TI-Nspire file, it's called a TNS file. This is what will play on the calculator or on the TI-Nspire software, Navigator software. There's always going to be a student worksheet. So if you're into having a hard copy of things, this is where the students could follow along on a piece of paper. Teacher notes are always available, and teacher notes kind of give you the answer to some of the questions that might be posed during the activity, and then you can download all the files if you'd like to. So this is like to introduce you, as Stacy said, virtually to this young woman. Her name is Dr. Kristi Egland, and she is a research scientist at a facility, as I mentioned, where I live. I live in Sioux Falls, South Dakota, and it's kind of a, believe it or not, I know some of you are going, yeah, right, but believe it or not, it's like a healthcare mecca in the Midwest. It's like a notch perhaps below the Mayo Clinic. We have two very large hospital systems and a very, very large research facility where Dr. Egland works. And her main area of research was and is breast cancer. And so we had the privilege of meeting Dr. Egland and interviewing her and talking to her about her journey and struggle with breast cancer. It turns out that Dr. Egland developed the exact same type of breast cancer that she was researching at the time. And so she was an expert on it. She didn't want to be an expert from the standpoint of having the disease, but she was an expert on it. I love this quote that she made, and this is so her. And she was so upfront with us. She said, I have a love-hate relationship with cancer. I love to study it and hate having it. And she was incredibly blunt about that. And she was so gracious with her time and so honest with her emotions with us about this whole cancer journey. So anyway, that whole process I just took you through is where can you find this activity? Education.ti.com, STEM activities, STEM behind health, and there it is. When good DNA goes bad. All right, so I'm going to share the activity now and move over to this. This is going to be showing the TI-Nspire CX Premium Edition teacher software. And this is, whoa, what did I do there? See if I can get that back up here. Sorry about that, guys. Okay, let me stop the share. We'll get it here. All right, hopefully everybody can see this activity now. So this software that you're seeing, the place where the activity is being shown is on that TI-Nspire CX Premium Edition software. And this is the splash page or the cover page of this activity. Now I want to be clear right up front. I mean, the title of this activity is, you know, a little bit condemning, almost like when good DNA turns into bad DNA. So like when DNA turns into a criminal or something. There's no such thing as bad DNA. I mean, I would argue that DNA is DNA is DNA, and DNA is made of pretty simple pieces and pretty simple components. But it's just that the way that DNA is assembled is what can make it either normal and beneficial, or it can go rogue. DNA can cause, I should say, rogue things to happen in cells. Now, DNA quick little lesson. I don't want to insult anybody and I don't want to talk down to anybody at all, but it may have been a while since you've had a biology class. And I'm not going to go into this in great detail, but DNA, most of your DNA is housed inside the center part of your cells, which is the nucleus. And almost all of your cells have a nucleus. And in your nuclei of your cells, because you are a human, because we are human, we have 46 pieces of DNA. And those pieces of DNA are called chromosomes. So chromosomes is a word that we throw around a lot. Most everybody's heard of them and so on. But the DNA that makes up a chromosome looks like a very long ladder. And that ladder is twisted into this thing, this shape called a double helix. So what DNA does, I always told my students, because I taught biology, DNA really doesn't do anything. DNA basically sits there in the nucleus and allows itself to be manipulated. And that manipulation involves a process that ultimately will produce what DNA is designed to produce, and that is proteins. So the manufacture of proteins is what DNA's job is. So if DNA goes bad, well, let's go to the first scenario. If the DNA is air quotes good, that means that DNA is producing proteins that it was supposed to produce. If DNA goes bad, that means that some of the proteins that it was designed to produce either don't get produced or they get produced incorrectly. And that is what can lead to some problems. In fact, cancer can be and usually is the result of DNA not doing what it's supposed to do, or not coding for what it's supposed to do, or supposed to code for. This whole coding thing got to be way more commonly understood when people started actually doing coding on a calculator or a computer, or for like a computer science class, because the code doesn't really do anything. But what the code says to do is what causes the result. Okay, so I hope that kind of clears that up a little bit. And I don't want anybody to go, oh man, I hope my DNA is not bad. Well, your DNA is not bad. Your DNA is just DNA, which is allowing itself to be manipulated. All right, so a lot of this activity, in fact, are activities that are on the STEM Behind Health website are like human interest stories. I know that doesn't seem real mathy. I know that may not seem yet real sciencey. It may seem kind of fluffy, but believe me, this is not fluffy at all. This activity, as I mentioned, Dr. Egland's a star of this particular one. My former student Chelsea is a star of the type 1 diabetes one, and it's real people. This is not textbook hypothetical stuff. These are real people who have had real conditions and how they dealt with them and what caused them. So Dr. Egland, as I mentioned, a breast cancer scientist, it's not Stanford research, it's Sanford without the T. She was diagnosed with, and there's that first piece in red, stage 3 triple negative invasive ductal carcinoma. Now that's a mouthful. That's just another way of saying a type of breast cancer. And to date, I believe there's about a dozen different types of breast cancer that have been identified. Not all of them are as severe as others, but this particular cancer that Dr. Egland had has 2 parts to the name that cause alarm. The first one is right here, stage 3. Cancers are typically, at least breast cancer, is typically measured on a scale of 0 to 4. So stage 0 is like brand new. I mean, the cancer was just picked up and diagnosed very recently. And I think it goes without saying that the smaller the stage number, the more likely it is that that can be treated and cured. Stage 3 is one stage below the top stage, which is not good. Stage 4 tends to be a little bit more difficult to manage. This is the other part. And if you know somebody, or maybe you yourself have had to deal with breast cancer, when you see those two words, triple negative, these types of breast cancers tend to be the more difficult type to, not to diagnose, but to treat. And the rate of remission, rate of recovery, rate of survival tends to be a little bit lower with a triple negative type of breast cancer, especially like Dr. Egland's situation. She was in stage 3. And keep in mind, if you missed this before, this is the exact type of breast cancer that she was researching when she was diagnosed with it. So there's some other words here: invasive, that never sounds good. Ductal, we'll look at that in a little bit. That has to do with the ducts inside the breast tissue. And here's this suffix right here is one that many of you have probably heard in other realms. Now, Stacy, I know you're monitoring the chat, so I don't know if people want to throw something in the chat, but are there other diagnoses that you may have heard of or that you may have received that have the word OMA at the end, or maybe N-O-M-A? And I know I have. I have had several bouts with skin cancer. And if you could see me now, you go, yeah, that makes sense, because I'm like very, very lightly pigmented. And when I was a kid, I thought I'm going to be Mr. Tanning guy, never was. And so now it's kind of catching up to me. But the type of skin cancer you really don't want to hear that you have is melanoma. Okay. We also have basal cell carcinomas and that type of thing. But that NOMA typically means it's some sort of tumor, some sort of cancer. So this was in June of 2007. So we're talking about 15 years ago. And this was chilling to me when I first asked Dr. Egland, what's the first thing you thought when you heard you were diagnosed with this stage 3 triple negative? This is exactly what she said to me, you guys. My first thought was, I'm going to die, because she knew what that meant. And I'm going to give you a little spoiler alert so you don't get, right now all depressed and say this poor woman, yada, yada. She ended up recovering from this. And I know I should have saved that happy ending for the ending. But I want to let you know that even going into this webinar tonight, when we're talking about something that can be kind of grim, there is hope. And she would be the first one to say, there is hope. Don't give up. Because she was not too far from having no hope. So what we do for the students here, and these are designed for the students to use. I mean, I love it that we have adults on here and they're tuned in and want to learn about this. But students need to learn about what words mean and what they don't mean and what they're not. Tumors, okay, they've all heard of that. Mammograms. Chances are a student, even if it's a high school young lady, she may not have had mammograms yet, but her mother or somebody in her family may have. And then another word that's on here is a biopsy, where just either with a needle or with a scalpel, a little piece of the suspected tumor is removed and analyzed. And Dr. Egland, right away, when she saw this tumor, she saw this in her own mammogram. And when she saw it, she was kind of hit to the core, because she knew what it meant. Again, this is Dr. Egland. Again, delightful young lady. To me, everybody's a young lady, because I'm not very young anymore. But she was a wonderful person to work with. There's I think a huge underestimation, perhaps, or a misunderstanding about what cancer is and what cancer isn't. First of all, let's talk about a little bit of the DNA of this. And this is going to sound kind of strange to some of you, maybe to all of you, but I just want to make a statement that you may not have ever heard before, and that is this: all cancer, ALL, all cancer is genetic. And I'm going to just let you think about that for a second. All cancer is genetic, but some cancers are hereditary. There's a difference between genetic and hereditary. Genetic means it involves genes. genes are pieces of chromosomes. Chromosomes are made of DNA. So all cancer has its root cause in the DNA. Something changed the DNA, something caused a mess up in the DNA, and so that resulted in the development of cancer. Not all cancers are hereditary. Now, breast cancer, everyone's, I think, pretty well aware that breast cancer indeed does have, at least some types of breast cancer, have a hereditary component. That's why when physicians ask for health histories, especially for women, when health histories are being sought for patients, they talk about, is there cancer in your family? And breast cancer is one of those that does have a hereditary component, but it still all goes back to the DNA. And we don't have time to, and nor do I have the expertise to talk about all the things that can happen in the DNA that can cause cancer. I just know that, to me, it's absolutely incredible that, with all due respect to any of you who may have experienced cancer or dealt with others who have experienced it, it's amazing to me that cancer doesn't happen more often. And the repair mechanisms that are going on within our cells are phenomenally efficient. So cancer is basically this: abnormal, whoops, get the A in there, abnormal cell growth or abnormal rates of growth. A tumor is a ball of your own cells. This is something that people don't think about very often. Those cells are yours, and they just went rogue. They just got out of control. They started growing at, as it says here, an uncontrolled rate. Now, what that implies is your cells typically grow at a controlled rate, and they are under very, very strict control. But when they get out of control, that control usually is messed up because the DNA got goofed up somehow. Now, you're waiting for me to use maybe biological words. We could use the word mutation, perhaps. A mutation within the DNA changes, which is going to result in a changed protein or something like that. All right, so the challenge then with cancers is to either prevent normal cells from developing into the ability to divide and control it. So in other words, prevent cancer or stop cancer. Now, I think you can think of ways, kind of brainstorm in your own mind, how do you prevent yourself from getting cancer? Well, I didn't. I was in the sun a lot, and ultraviolet light is a known DNA messer-upper. I don't think they call it that in textbooks, but that's what it does. It messes up DNA. It changes it. But I didn't pay any attention to that. We all know that there are other things that you can put into your body or onto your body that have the potential to cause DNA to be changed and goofed up. All right, let's keep moving. When we get to this activity, now I always get people ask me about these activities, like with the zombie activity, for example. What do you think the age of the kids should be that use this? Because there's some pretty scary stuff in the zombie activity. And this particular page in the breast cancer activity may not be something you want to show your students. I don't know. I mean, I don't know your students. I know in high school, I would say that this is mostly appropriate. I taught AP biology, and when I did this activity with them, you always get your one or two students that might snicker about it or make a joke about it, but you as a teacher and you as a professional know how to put a stop to that. Once they see what this actually is showing, I think they develop a little bit more appreciation of it. So basically what we have here is between you and me, kind of a fake mammogram from the side. And right here, I don't know if you can see it, but right down there's a little green dot, like this fluorescent green dot that is supposed to represent a cell. Now, we all know cells are not that big in relationship to the rest of the body, but I'd like you to see what's going to happen here. We have a scoreboard, cancer cells. We have one cancer cell right now, and our time is, let's say, today. So we're at time 0, okay? So I'm going to click on the play button. If you've never seen a simulation on TI-Nspire, you don't expect a calculator to do what this calculator can do. It can have animations, and it can run, as you'll see just in just a moment, a simulation. So I'm going to click on the play button, and we're going to take this through. You can see here 365 days, which is a year. And it's not going to take that long because we're going to go flying through this year. It's going to be like fast forward, time lapse photography. So you can either watch up here, actually you can watch both places, flip flop back and forth, the scoreboard, and you can watch what happens with this tumor growth. Okay, so I'm going to go ahead and start this. And this is a simulation of what a tumor growth may look like. And we're winding down now. There we go, 365 days. And now you don't have to look very hard to see that fluorescent green blob or mass in the lower part of the breast tissue. By the way, if you're trying to get your perspective, this person would be facing to my right, to your right. These, and we'll see this in a little while as well with a colorized version. These are ribs right here and muscles and the breast tissue right here. I don't know if you were paying attention, but Dr. Egland, when she saw her mammogram, the tumor that she saw in her breast tissue was about 4 centimeters in diameter. 4 centimeters is about 1.5 inches. So we tried to make this about realistic to that. And 1.5 inch tumor is pretty good size. But if you check this out, and I don't know if you're watching what was happening with the numbers here, and this is a great example, if you're a math teacher, of exponential growth. Because if you think of how cells would divide, if you started with 1 and it divided, you'd have 2, and then if they divided 1, 2, 4, 8, 16, and so on, and you double the number each time, your rate of growth is not going to be linear at all. I mean, it's going to be exponential growth. Now, along the way, do some of those cells die? Probably. But what does this number mean? 4.22 or 212 E plus 0.9. So the way you would read this is 4.2, I'll round it off, we'll take the liberty of that. 4.2 times 10 to the 9th. 10 to the 9th is a billion. So in 365 days, in our simulation, this cancer went from 1 rogue cell to over 4 billion cancer cells. Now, it seems like a year is a long time, but that rate of growth is pretty phenomenal. Now, if you wanted your students to run this again, you could reset it. These buttons, if you've not seen a simulation, are kind of the same as they would be on YouTube. You can reset it, start it again, and so on. So I would encourage you, if you want to do that, go on the TI website, download the activity into your software, and take a look at it. A couple other terms that we want to make sure the students understand. Not all growths are deadly. I mean, when we hear the phrase cancer, or we hear lump or tumor, that a lot of people think, well, that's a death sentence. And even more today than 10 years ago, it isn't necessarily. And there's 2 main kinds of growth that happen. This word is pronounced benign. You know that, your students may not, because there's a G in there that we don't pronounce. A benign tumor basically means it's a contained mass of either cells or fluid or something like that. The M word is the one where it's a lot more serious. Malignant means that the tumor is potentially able to spread to other parts of the body. And that spreading process is called, I've heard it pronounced a couple different ways. I prefer metastasis. There's another way you can say metastasis. I've had people actually get offended when I say this because they don't listen to the rest of my statement. Breast cancer itself is not fatal. Let me finish. The fact that the tumor is in the breast tissue, to my knowledge, is not going to cause somebody to die from it. The issue becomes if the cancer cells from the breast tissue spread to other parts of the body. That process is called metastasis, and that would mean it would be malignant. This is why when people have breast cancer surgery, like mastectomies or whatever, the physician, the surgeons often explore and remove lymph nodes around the breast tissue, like under the arm and so on, because the lymph nodes are part of your circulatory system, sort of, that the cancer cells can be spread with the lymphatic system. All right, let's move on. Throughout this activity, as with most TI activities, there are questions that are embedded in it. So as the student goes through these activities, there's kind of these spot checks along the way. This is talking about a cyst, and cysts are typically benign. So the student would answer that way, hopefully. We have a little bit of anatomy lesson here, and I'll just go through this pretty quickly. This is pretty much the same image we had before. Now it's in color again. Age appropriateness, that's up to you. I'm not going to make that call. That's completely up to you. If you're going to use an activity like this, is it possible to not show these pages? Absolutely. You can take these pages out of these activities. You can delete this from the activity if you choose to. If you feel like you need to get parental permission to show things like this, that, again, totally up to you. But if not, if a kid's in high school, especially if they're in an anatomy class, I think this is very appropriate. Now, one of the cool things here, whenever you see a magnifying glass in a TI activity or an Nspire activity, you can grab that by the handle, and then you can move that over to this area, and wherever a little question mark pops up, oh, look at that, there we have one. You let go of the mouse, and it says that's a rib, and the rib's attached to the sternum, and so on. So a little anatomy lesson. Let's go back here. We have blood vessels, which is good and can be potentially bad, because if you have a metastatic tumor, the blood, along with the lymphatic system, can carry tumor cells from one place to the next. This is, if I can get to this area here, there are the ducts. The ducts are toward the front part of the breast itself. The job of the ducts is to carry milk from the lobes out to the nipple so that the baby has something to eat, has milk to drink. These are the lobes. These are the ducts. If you remember, Dr. Egland had triple negative invasive ductal carcinoma. So her tumor was located in that duct area. Okay, let's move on. So then we talk about where's the milk produced. And here's Dr. Egland. We're going back to her. Where do you think her cancer started? And it actually started in the ducts of her breast tissue. This is another lady that we had an opportunity to visit with. And the reason we put this in there is not so much for you folks, but for your students. This whole business of cancer diagnosis, treatment, surgery, and whatnot is not done by one person. I mean, I know there's television shows about medical professionals that pretty much have them doing everything themselves. And my experience has been that is not the case. This is another person, Anu Gaba. Dr. Gaba was involved in medical oncology, and she's also a researcher, but she's a practitioner as well. And she works very closely with Dr. Egland. Now, this is, if you're a math person, and statistically, some of you probably are, because a lot of math teachers end up on these webinars, which is awesome. The rate at which cells divide and the number of new cells you make every day is staggering. Now, it says here, you make as many as 100 billion new cells every day. Now, at my age, and this is kind of age dependent, at my age, I'm probably not making nearly that many anymore, because the older you get, your rate of cell division goes down. You don't make as many. That's why sometimes as people get older, they appear to shrink, they get a little smaller and so on. But imagine this. Your DNA is made of 3 billion rungs of that ladder I was telling you about, 3 billion of them. And every day you replicate all of them as many as 100 billion times. That's those two numbers. I mean, we think we can understand a billion. I would argue that most of us, including me, I can't get my head around what a billion is. That is a gigantic number. So the fact that your cells replicate, which means make an exact copy of all of those pieces of DNA billions of times a day, is incredible to me. And I taught biology, so I should be like, yeah, ho hum, whatever. The more I taught biology, the more staggering it became to me, how miraculous biology was. Okay, so I'm going to totally put myself out there and I'll embarrass myself. And since you're all muted, you go right ahead and laugh at me because I am pretty bad at this. But we're going to play a little game, and we have the directions, but who are we kidding? If your students are like my students, they don't read them. So we just go to this DNA replication game. There's 3 speeds. We can go slow, medium, and fast. If you really want me to be embarrassed, we would go fast, but we're just going to go slow. So what's going to happen, a little spoiler alert here, when I click on the play button, this DNA, this is the ladder, these are the rungs, so like this A and T and this G and C are little rungs of the ladder, and here's the sides of the ladder where the 5, that little mark that looks like an apostrophe is a prime, 5 prime and 3 prime. This is going to separate, it's going to pull apart, like we're going to yank the ladder apart and expose the rungs. And when we do that, my job is going to be to try to fix it, to get it back the way it was by putting in new letters to replace the ones that are already there. So check it out. G and C always go together, and A and T always go together. So let's see how I do with this. You know, I've been practicing night and day for weeks for this webinar, so I don't totally embarrass myself. So let's play it, unzip. So there's my A, the T came in there. Let me try to get a T in there. Oh, I missed it. I'm going to click on the T, that worked. I'm going to go with the G for the C. Are you laughing at me yet? Because if you are, I don't like you anymore. Don't make fun of me. Did I get my A? Okay, next is G. The next one is an A. You see how I'm doing this? This is a great lesson for kids because they invariably goof up somewhere along the line. You know, I feel pretty good about that. I think that's it. It just does the horizontal part. I had a replication error rate of 8%. So I missed 1 out of, I don't know, whether 12 or something like that. Let me just tell you this. If you have a replication error rate of 8% when you replicate your own DNA, that's not good. That is not acceptable. Let's put it that way. Just for kicks, I want to show you what it looks like when you go fast. And I'm not going to even try because I've seen this and I know that it goes really, hopefully I can click on it. Did it go there? Maybe not. Let's try to reset it. If we can't, we can't. If we can't, it's being merciful. I mean, all right, here's fast. Now watch how fast this one goes. And you're going to see why we do it this way. Because in real life, this is more realistic, but not even close to really realistic. Split, boom, 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 boom. I couldn't even find the letters that fast. You see how I'm goofing? I don't know why I'm getting some right. Maybe it's just like a courtesy score or something like that. So I had a 50% error rate there. Now here's why we do that. Why we show that particular game. Not only is it cool, the kid can, it's interactive for the kid, but check this out. Remember what DNA does. DNA codes for the production of proteins. Proteins are, you know, muscle is made out of protein. Enzymes, you've probably heard of insulin. Insulin is a protein. Anything that's pretty much hormone, or virtually every hormone is protein as well. Starts with DNA. And with 6 billion bases to replicate before each cell division, remember how many cell divisions you have a day? Like 150 to 100 billion? Mistakes will happen. It's inevitable that mistakes will happen. Thankfully, this is the miracle of what happens. Your cells actually fix those mistakes, hopefully. But think about this. The more mistakes that are made, the harder it is to catch them all. So if you do what I did, deliberately, without even really knowing it, deliberately put yourself in a situation where your DNA gets screwed up by sitting in the sun, ultraviolet light beating down on a milky white shoulder, over and over and over and over, day after day, year after year, the chances of having DNA that gets goofed up or mutated goes way up. And as a reminder, if DNA gets goofed up, the problem is seen right there in the protein that is either made or not made by that particular part of DNA. So here's just a little biology lesson. Which of the following is an incorrect pairing? I'll tell you right now, A's and G's don't go together. C's and G's and A and T's go together. That's very, very precise and very specific. If there's a mutation in a sequence of DNA, this is what we're looking at here. The protein structure may be altered or changed. So the protein is goofed up. Now, here's what's amazing to me. Let's look at this question. And if you're a math junkie and you like big numbers, you'll really like this. When your DNA replicates prior to mitosis, is just a type of cell division that makes new cells. There are about 6 billion bases, that A, T, C, and G's are called bases, that must be exactly copied and put in the right place. 6 billion for every cell division. If you replicated 100 bases per second, how long would it take to replicate all of the DNA just one time? All right, I'm going to save you a little bit of work. Now, some of you are probably frantically pecking away at a calculator, or if you can do that in your head, rock on. I don't know very many people that could. Let me give you the answer. If you did 100 per second and you had 6 billion to do, it would take you 700 days to replicate your DNA. And yes, I did calculate that. I'm nerdy. I thought, I wonder how long that would take. And I figured it out, 700 days. So if in my mind, 700 days is like 2 years. It would take you 2 years to replicate your DNA one time. So some of you are like, well, how do we do it? Well, the way we do it, if you go back here, I'll give you just a really 10-second lesson on this. This is the DNA that got pulled apart. What you really do is you pull it apart in many, 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 many places and replicate bunches of places at the same time. So it really speeds it up. Otherwise, there's no way you couldn't replicate your DNA once every 2 years on one cell and expect yourself to survive. Okay, now here's where a life lesson for the students might come in. And we all know that teenagers think they're immortal. They think that won't happen to me, and so on. But this is kind of that, okay, now I'm going to give you the talk here, guys. This is where your choices you make in your lifestyle, especially for a young person, if they start making good choices early, the chances of them keeping those choices pure and good are better. Smoking and chewing tobacco are responsible for about 30% of all cancer deaths. Now, I mean, there's no question about that. I mean, this is pretty significantly researched. So, cigarette smoke, one of the concerns I have with any kind of smoking is that you're exposing your cells in your lung to chemicals that are potentially dangerous. And those dangerous chemicals are called carcinogens, which is cancer causers. And there's no doubt that cigarette smoke is a carcinogen. So what does that mean? A carcinogen just can increase the mutation rate in the DNA or increase the change rate in the DNA so that the DNA gets altered to the point where it can cause problems. All right, I was going to mention something else, but I'm looking at the time and we're going to have to tough it out here. So let's get back to Dr. Egland. What did she do? Immediately, like within a week of her diagnosis, she knew what it meant. She underwent a double mastectomy, which is where they remove both breasts. She only had tumor in one, but she chose to have both breasts removed, had several lymph nodes removed, and check this out. She had 8 rounds of chemotherapy, which doesn't seem like much, but I've never had internal chemotherapy. I've had some on my skin, on the externally, topically. But chemotherapy is basically, we call it a medicine, it's basically a poison. And the intent is to poison cells that are growing fast. And it's getting more and more precise, but it used to not be. It used to be really indiscriminate, like go kill all the fast growing cells. That's why people's hair fell out, still falls out. That's why they get nauseous and so on, because the stomach lining is really fast growing cells and they start to kill them off. Radiation therapy is another very common means. And look what she had, 33 rounds of radiation therapy. Thankfully, the breast tumor that she had was fairly close to the surface, so radiation was definitely an option. So here's one of the things we try to do in these STEM activities is call out potential careers for students. And here we have the team that I referred to before. Dr. Gaba and Dr. Kristi Egland are just two of the team members. Look at all what went into this. Radiologists, surgeons, reconstructive surgeons, chemo, radiation, genetic counselors, nurses, lab tech. I mean, this is not you've seen maybe the TV show House. I mean, he's a great doctor, but he can't do everything. I mean, there's a way more teamwork than sometimes we give credit for. Cancer doctor, as you know, is called an oncologist. Some of the DNA that actually can get potentially messed up, if it can cause cancer after it's messed up, those are called oncogenes. Onco means cancer. Here are some of the treatments for breast cancer. Dr. Egland underwent many of them. Here's why, and I want to make sure I mention this, because if you remember back to what we were talking about before, and I'm going to probably end up having to wrap up with this, Stacy, so believe it or not, Stacy and I have worked a lot together. She knows that my timing is not good sometimes, and I don't care about time. But here we go. This triple negative, what does that mean? Triple negative. We know what the triple means. It's 3. Negative means there's receptors. There's these little satellite dishes on your cells. They're called receptors. They are proteins. And if a breast cancer is triple negative, that means it's missing 3 of those types of receptors. And the 3 that typically are missing are receptors for estrogen, which you've all heard of, a receptor for progesterone, which you've also heard of probably. And the third one is one called HER2, which stands for human epidermal growth factor receptor. Those 3 chemicals, estrogen, progesterone, and HER3, are growth-causing hormones. Now you think about estrogen and progesterone. Those are steroid hormones. And in order for a steroid to get into a cell, there's got to be a receptor on the outside. Now, why is this significant? Check this out. A lot of times, hormones are used as delivery mechanisms for chemo, or for some kind of a chemical to kill cancer. So you shoot these hormones into somebody, estrogen, and you link to it a chemical that will kill the tumor cell. Well, the only way that chemical is going to get into the tumor cell is if there's a receptor for the hormone. And if there are no receptors for the hormones, the chemical just floats around in the bloodstream attached to a hormone and doesn't have anywhere to go. I mean, it's phenomenally cool, but it's phenomenally nerve-wracking too. But hormone therapy oftentimes works very well. We have surgery, chemo, radiation therapy. One of the more recent developments is precision oncology, where the team figures out exactly what type of tumor it is, and then manufactures or designs a particular drug or treatment to go after that particular type of tumor. These are some of the side effects that Dr. Egland experienced. Incredible to listen to her talk about that. It was tear-jerking to me. The next page or two just talk about, well, if we do treatments, different kinds of treatments with patients, what happens to the tumor growth. We don't have time to do that right now, but basically the growth is different depending on the treatment we use. And let's wrap this up by getting into the end of the story. In spite of all of this that happened to Dr. Egland, she started to get some good news. And then she started to feel guilty. Like, why did this work for me and not so many of the other people that I was working with and researching? And to this day, she still feels sort of bad in kind of a funny way that she was cured and other people were not. All right. I don't know if I took a breath there that last 15 minutes, but that's the end of that activity. I would encourage you, if you don't want to do this with your students, if you have if you teach something where your curriculum doesn't really have anything to do with health and wellness and breast cancer, or whatever, like you're teaching a math class or something like that, then no, maybe this would not be something you'd want to yourself show your students in your class. However, chances are pretty good you do have science teachers in your school, and some of those science teachers surely are life science teachers like biology or AP Bio or anatomy and physiology. Share this with them, because I can promise you they would be interested in this, not only in the cancer piece of this, but in the biology of DNA and all that type of thing. Stacy, I came close, didn't I?
You did so good, Jeff.
Stacy's pretty forgiving, but anyway.
It's real. It's so hard when you have a webinar and you want to really share your experiences with the activity and your experiences with students or working with teachers and how impactful the technology is, to get it all chunked in about 50 minutes. So it was great. It was great. You did great.
Let me end with a comment I made at the beginning, and that was this. You don't expect a graphing calculator to be able to do what you just saw. And that's just the tip of the iceberg. I mean, if you go on the STEM activities and if you go into science and math inspired, especially nothing against the math activities, are great, but many of the science activities and science inspired have simulations that just blow me away, that you can actually watch a simulation moving on a graphing calculator screen. Those of you who said, there's no place in science for graphing calculators, I'm glad we're not together, because I'd probably stick my tongue out at you at this point, because there definitely is a place for graphing calculators, especially this one in science classrooms. Okay, Stace, I'm done.
No, you're good. So in the chat, I did post your certificate of attendance as well as any associated documents that Jeff might have shared for you guys. Like I mentioned earlier, this webinar is recorded. You'll receive a follow-up email in a couple of days with the certificate of attendance as well as the document. You will also receive a post-webinar follow-up. This really drives how TI looks at future webinars. But then also, if you have questions, you need somebody to contact you, you want more information about people like Jeff coming to your school, or just more information about the technology, please let us know. They really do listen. I'm still in the classroom. I call probably TI Cares more than I probably should, but they really do listen and have our best interest, because it's what's best for students. If you are interested in other webinars like you guys did for tonight, you can sign up for others. Jeff mentioned that he does have another one on zombies in October. My good friend Jessica and I are doing some STEM projects in November, if you're interested in doing some STEM with coding projects, kind of similar to what Jeff was talking about. Again, thank you, Jeff. I've used this activity. I taught anatomy and physiology. I also taught the Project Lead the Way curriculum, where we actually had a breast cancer curricular component. But also it allows students to see the real life application of things that might be affecting them, but at the same time, how a math science concept can be covered, using technology. So thanks, Jeff, for being with us. Thank you guys all for being with us as well tonight. And we look forward to seeing you on future webinars. Thank you.